thank you, all of you, for coming tonight. I think we're in for a great treat. My name is Khaki Hopkins. I'm president of the Dallas County Medical Society Alliance Foundation. For one more week. Smile on her face. And that brings me to the first of my introductions for the evening, the precious soul who is going to replace me.
I can't replace him.
Stand up, Rulon. Rulon was the mother of this house for 3 years. So I know if you've been here for the last 4 or 5 years, you have seen Rulon. And it was a great relief to me to know that I really didn't have to worry about the Aldridge house. She knew everybody. Everybody knew her. They all had her telephone number. They didn't know mine. So we were in great shape at my house. And then my second introduction. Many of you have already had the pleasure of a visit from her at your table this evening, but our extraordinary chef, Wendy Henson. She insists she is not a chef. She's just someone who loves to cook. And aren't we glad? Yes. She is actually on the board as the chair of hospitality. Oh, she do it well. Well, our staff has made wonderful progress this year, in spite of the fact that we were all kind of cocooned in our separate homes. But as everybody else in the world figured out, there is a way to communicate other than person to person. We had some introduction of new concepts and some great new projects. And the one that thrills me the most is the Alliance Retired Physicians Project. It was a dream. And it actually, I have to tell you that it happened because my husband retired. And I thought, mercy, he's a good volunteer. He was out there volunteering, teaching English as a second language with, used to be Victory Meadow, Literacy Achieves. That's right. Who told me that? Who said that?
That's right.
Thank you.
I was a teacher.
Hey, and Shelton was back there saying, haven't you learned the name yet? At any rate, it was a great hint that he gave me that life after medicine doesn't have to be boring. It can be filled with wonderful service to the community, and that's sort of why you guys are here. Because one of you was a good example. And then I met another wonderful example when I met Marvin Stone. I cannot tell you how much I see that this new group of men and women who spent their lives in medicine, not all their life, the first part of their life in medicine, has enriched our membership tremendously. That infusion of new talent, new skills, is exhilarating to this organization. And it has caused the most incredible lineup of programs this year because of their newness to us and the fact that they could bring what actually brings us all together as a family of medicine anyway to us in a different way. And the board joins me in thanking all of you for being a part of our inaugural class of Alliance Retired Physicians. Thank you. Of course, you know the man most responsible for bringing all this into fruition. It is our wonderful friend, Dr. Marvin Jules Stone. He's known to most of you by his many accomplishments. But I want to tell you a little bit about his trip getting here. He was from Ohio, he's a native of Ohio. He earned his MD with honors from the University of Chicago. It's kind of nice. And then he did his residency and fellowship at Barnes Hospital, the National Institute of Health, and UT Southwestern. He's the first chief of oncology and director of the Salmons Cancer Center, Baylor University Medical Center, Dallas, for 32 years. He's a master in the American College of Physicians and a fellow in the Royal College of Physicians, London. Currently, he's chief emeritus in hematology and oncology at Baylor, professor of medicine at Texas A&M College of Medicine, and a clinical professor of humanities at the University of Texas at Dallas. It is my pleasure to introduce to you my friend, our friend, Marvin J. Stone.
Thank you, Kathy. Even my mom could know. Thank you all for coming. Thank you for being here. We have a real treat for you this evening, I think. Before I forget, though, I do want to thank Brittany Livling, who is in the back there, who is the communications director for the Southwestern Medical Foundation. She and Kathleen Gibson, the president and CEO, were able to give us the wonderful booklets that are on your chairs. That was published about a year and a half ago, and is a marvelous summary of what you're going to hear about, including Dr. Olsen, who's got an article in there himself. So anyway, thank you so much. We appreciate it. I'd like to tell you a story before we get into the main program. I like to tell stories. And this one is about an overnight train ride. And on the train that night were two people. One was William Castle, who was a Harvard very well-known hematologist. And the other one was Linus Pauling, who was to get his Nobel Prize for chemical bond, nature of the chemical bond, about 5 years later. And they were chatting as the train was going, and Castle told him about this unusual disorder called sickle cell anemia, and how the cells change size, particularly with low oxygen tensions. And Pauling listened and thought, well, there must be some structural change in the hemoglobin. And hematologists like to look at blood smears. So I'm going to pass this around. You can see sickle cells. But when Pauling got back to Caltech, he got some of his clinician friends to get some blood on some of their sickle cell patients. And he performed a series of studies, and one of the experiments that he did was to show that there was a difference in the hemoglobin migration in electric field and electrophoresis between normal hemoglobin and sickle hemoglobin. And that led to a publication in Science in 1949. And it was called Sickle Cell Anemia, a Molecular Disease. And it should be noted that this was 4 years before the Watson-Crick paper on DNA structure. So this was really a landmark paper. And one of the things that came out of the study of sickle cell hemoglobin, besides Pauling's observation, was the fact that these cells that became banana-shaped or crescent-shaped became rigid, and they obstructed blood vessels. And when they obstructed blood vessels, all kinds of trouble happened because of wherever the blood vessels were occluded. But in addition to that, it was associated with very severe, sometimes excruciating pain. And anyone who's taking care of patients with sickle cell pain crisis doesn't forget that. So sickle cell anemia has been with us for a long time, and that has been known for a long time. But one of the things that makes it so exciting now is what you're going to hear about from Eric Olsen about the CRISPR technology, because sickle cell anemia may be one of the best candidates for gene editing. With that, let me tell you a little bit about Dr. Olsen. We're so glad to have him. He was educated in North Carolina at Bowman Gray and got a PhD in chemistry. He had postdoc work at Washington University in St. Louis, and then was at MD Anderson for several years until 1995. And that's when he came to Dallas as the founding chairman of the Department of Molecular Biology. He holds 3 different named chairs as head of the Department of Molecular Biology. He's a member of the National Academy of Sciences and the American Association of Arts and Science. He and his colleagues have discovered many key genes and mechanisms responsible for development and diseases of heart and other muscles. He's received a number of awards and prizes and co-founded multiple biotech companies. Listen to this. He's among the most cited scientists in the world. His work has been cited over 100,000 times. Now listen to this. He plays in a rock band. The rock band is called the Transactivators. And he plays guitar and harmonica. I don't know, did you bring any of those?
I left it at home.
And all this was inspired by Willie Nelson. And I think Willie Nelson supports your research. But what we're going to hear tonight is a new era in biology and medicine, a new strategy for correction of a different disease, Duchenne muscular dystrophy, using CRISPR gene editing. Eric, thank you so much for
Thank you all so much for coming out. Really happy to be here. I'm happy to be anywhere after being on Zoom calls for a year. No, it's great. I've enjoyed meeting many of you. Actually, my wife's a real doctor. She's a retired pediatrician, so maybe she could join your organization. And one thing that Marvin said, so it's true, Willie, I know Willie Nelson, and he supports some of my research, and so I've spent quite a bit of time with Willie on his tour bus and on his ranch. People always ask me, what's it like on Willie's tour bus? And I say, I would tell you if I could remember. So, yeah. I'm really happy to be here. It's kind of hard to give a talk on molecular biology after dinner, but I know we got a lot of smart people here, so maybe we'll have a quiz at the end. So I want to talk to you about CRISPR. How many people have heard about CRISPR? Well, that's pretty good. You'll all be ready for the test questions at the end. So I want to talk to you about the most revolutionary technology that has come about probably in the past century, and has for the first time has offered the opportunity to change the blueprint of life, not just the blueprint of human beings, but of all living organisms, plants, and animals. And this is already revolutionizing biology and medicine, and the things that CRISPR will do will ultimately exceed all of your and our imaginations. So how did we get here? My lab has worked for 40 years on the mysteries of muscle. And why muscle? To put our work in context, muscle provides the meaning meaning to life. From the beating of the hummingbird wing, which can beat 100 times per second, to the movement of the largest animals that ever roamed the earth, to the beat of the heart, beats 1 time per second from embryogenesis to adulthood and defines the span of life, to the intricacies of the more than 500 muscles that form the body, to the precision of music. Here's Willie at a recent concert prior to the pandemic, to the intricacies of music and the power of athleticism, the 90-mile-an-hour fastball. All of this occurs through the power and the precision of muscle, and it requires incredible sophistication of biology to create these tissues. So over the last almost 40 years, members of my lab have sought to discover the genes and to establish a blueprint for how all of these muscle tissues form. Skeletal muscle, which forms 40% of your body mass, the heart, and all of the smooth muscles that form the vasculature. And my students discovered most of the key genes that form essentially a molecular blueprint that tell the body how to make these muscle tissues. And this is an ancient blueprint that exists all the way back into primitive insects, flies, all the way to human beings. And along the way, it became apparent that many of the genes that we were discovering were not only important for making these muscles, but they were also important for malfunction of these muscles, and that these genes were really at the heart, no pun intended, the heart of muscle disease. And so building on that, together with my trainees, I was able to found 4 biotechnology companies that are summarized here in the steps in the pathways of muscle disease in which those companies were focused are illustrated. And what I'll do today is I'm going to talk to you about our efforts through just one of these companies called Exonics Therapeutics, which has worked to develop a completely new therapy for one of the most devastating diseases of mankind, known as Duchenne muscular dystrophy, using CRISPR gene editing. So I know you're all not, I was told there's not too many molecular biologists in the audience, so I'm going to start with some basics here. Person, animal, made up of cells and tissues and cells, and then there's chromosomes, and then there's DNA. So the human genome is the blueprint of life. And the human genome contains 3 billion letters of DNA packaged into chromosomes, and it forms a double helix that you're all familiar with. And these 3 billion letters of DNA encode 20,000 different genes. Those genes encode proteins, and they're expressed in different cell types in your body, from muscles to nerves to the liver to the skin to the eye. And whatever combination of genes is expressed in a cell, that dictates what kind of cell that becomes. And this is, I don't want to get into too much detail, but this is important for what we'll talk about. This is the central dogma of molecular biology. This is the dogma upon which all biology is built. You start with DNA, that's the blueprint. It encodes all the instructions for life on this planet. And that is transcribed into what's called messenger RNA. That's a single-stranded sequence. And then that encodes proteins, which are generated through a process called translation. So we're going to talk about that today. So the human genome, it's more than just 23andMe or Ancestry.com. It is literally the molecular blueprint for life, and there's 3 billion letters, and 3 billion letters is a lot of letters. If you want to know how many letters 3 billion letters is, it's about as many letters as 1,000 Bibles contain, and all the words in those letters. And even one misspelled word, one misspelled letter can cause a devastating disease, and I'm going to tell you about one of those today. So we work on muscle, as I've told you. There are more than 800 diseases in human beings that are caused by an error in DNA and that can lead to loss of muscle function, atrophy, or destruction of muscle tissue. These are due to mutations in these genes, and there's not a single cure for any of them. So I'm going to tell you about the most devastating one of all. It's really the Mount Everest of human diseases and muscle diseases, and that's a disease called Duchenne muscular dystrophy. It's abbreviated DMD. It was discovered in 1860 by a French neurologist named Dr. Guillaume Duchenne. And all the physicians in the audience will understand how this disease was first diagnosed and how it's still diagnosed today. It's diagnosed by what's called the Gowers sign. And typically, and I get emails and letters every single day from mothers all around the world, and the letters are usually pretty much the same. Their son, this is a disease that affects boys, their son, they notice, is having trouble, usually around age 2 or 3, getting up the stairs or keeping up with playmates on the playground, and they ultimately take them to the physician. The physician asks them to get down on the floor on all fours and to stand up, and their legs are so weak they have to use their arms to stand up. And that's the first sign of the Gowers sign. And then they get subsequent genetic testing, and the diagnosis is confirmed as Duchenne. So it affects 1 in 5,000 boys. It's the most common fatal genetic disorder diagnosed in childhood. And as we'll talk about, there's no cure. This is how the disease progresses. So the boys are typically identified age 2 to 3 or so with walking problems and weakness. No person that's ever been diagnosed with Duchenne has escaped this series of clinical consequences. It progresses to the loss of ambulation at a really sensitive time in their lives. These boys become confined to wheelchairs. Usually in the early teenage years, they lose the use of their arms. The diaphragm, which enables you to breathe, is a muscle, and the diaphragm disintegrates, so they have to have ventilation. Then their heart disintegrates because it's a muscle too, and then ultimately death ensues by around age 20 or so, 25 or so typically. So this disease is caused by errors in the DNA, mutations in the largest gene in all of our genomes. It's a gene that codes for a protein called dystrophin. I won't get into too many details, but I need to get into a few here. So the dystrophin gene contains 2.6 million letters of DNA. This is a massive sequence of the DNA that encodes this protein, and it's on the X chromosome. All males have one X chromosome. All females have two X chromosomes. And here's one of the challenges. Thousands of mutations are identified in boys with Duchenne. And so every day, typically when a young boy is diagnosed through molecular biology now, where they can sequence the region of the gene and they tell them what their mutation is. So they always write me and they say, my son has this mutation, can you correct our son? And I have to say, yes I can, no I can't, or maybe I'm not sure. And I'll tell you about how we figure that out. So why does this disease affect boys only? It's because, as I said, all females have 2 X chromosomes and boys have 1 X chromosome, and the gene that causes this disease is on the X chromosome. So the disease is passed on from women who don't know that they have a mutation in one of their genes, shown here in red, because they have another one that's normal, so that compensates for this one. But if this couple has children, any son that inherits this mutation from the mother, shown here, he will get it. Every time, 100%. Half of the boys from this couple will not get it because he'll get the normal X. And then half of the girls from this couple will be a carrier to the next generation. So the problem is that women are typically asymptomatic and they don't know that they're carrying the mutation until they have a son that has the mutation. And then once they have the son with the mutation, there's all sorts of family, they have to go through a lot of family issues to figure out who else might or might not have the disease. So here's the human musculature. Your muscles in your body constitute 40% of your whole body mass. It's the biggest tissue in your body, and it's comprised of muscle fibers. And I won't get into all the details, but the dystrophin protein that's encoded by this gene, it sits right underneath the membrane of the muscle tissue. And you can think of it like a shock absorber. It holds all your muscle tissues intact. Your heart's beating once a second, and if you don't have this protein, it starts to disintegrate, and all the muscles, when you're flexing them, if you don't have dystrophin, the membranes leak, and that's why these boys lose the ability to walk. And here's what it looks like. So this is a histological section that we would make through a muscle tissue, and it's stained with an antibody, and you can see the dystrophin protein in green. It's underneath every muscle fiber. Really beautiful architecture. And here's muscle from a Duchenne DMD. This is a mouse, but a human would look the same. It's missing the protein. And here you can see the beautiful architecture of normal muscle. And here you can see what happens when you get DMD. The muscle disintegrates and all sorts of fibrosis and fat infiltrates it, and the muscle doesn't work. So this is my most complicated slide, and then I'll get away from some of the molecular biology, but I need to show this. So this is the dystrophin protein, and just think about it as a shock absorber. It's got a bunch of rods in the middle that are like coils on a spring, and they connect it to the membrane. And the protein is encoded by the DNA, and DNA is segregated into pieces that are called exons, and they splice together like pieces of a jigsaw puzzle. You can see how all these exons fit together just beautifully, seamlessly, and they form they code for each of the protein segments that you can see here, color-coded. And this dystrophin gene has 79 of these exons, spanning 2 million bases of DNA, and they have to splice together just perfectly, seamlessly to form the dystrophin protein. So there's 7,000 mutations so far that have been identified in this gene worldwide, and they cluster in these regions right here in red. So almost all the mutations cluster there. So when I get an email, I often show them to my wife because she's a pediatrician, and they're so poignant. But they'll tell me, okay, my son has a mutation in exon 52, can you correct him? And I'll look at the map and I'll tell them maybe I can correct it or maybe I can't, and I'll tell you how we can do that. So here's the dystrophin protein again. That's the scaffold for the muscle, and it's like, you can just think about it like a shock absorber on a car. And there's a mild form of muscular dystrophy. The clinicians here will all know about this. It's called Becker's muscular dystrophy. Patients with this form of muscular dystrophy can live to be 70 and still walking around, not confined to a wheelchair. It's a really mild form of muscular dystrophy. And the reason that is because they're missing certain segments in the middle of the protein, but the rest of it's intact. And so basically they have like a shortened shock absorber. It'll get them where they want to go, but it's not quite as good as the normal one. So why is this disease has defied everything that people have tried to throw at it. And why is that? There's a lot of problems, a lot of challenges. The size of muscle, 40% of your body mass. You got to correct this gene everywhere. You can't correct it in one cell. You got to correct it everywhere, and you got to correct it in the heart. And the size of the dystrophin protein is huge. And there's thousands of mutations. And you have to get it corrected in muscle and heart, and you have to correct it for your whole life. Because muscle lasts for your whole life. Your heart, some of you may or may not know this, but the heart cells that are beating in your body right now are the ones you were born with. Your heart cells live your entire life. They never turn over. So I'm 65 years old. My heart cell's been beating once a second for 65 years. And so this is, once you make a correction, you got to correct it forever. So for the clinicians, these are some of the therapies that have been tested to try to correct Duchenne muscular dystrophy in boys. And it is so poignant because these are young boys and they're struggling and their families are completely emotionally destroyed. And all these things are being tested in them and nothing has worked. So we decided to try to do something that doesn't treat the symptoms of the disease, but it treats the cause of the disease. The cause of the disease is an error in the book of life. It's an error in the gene that codes for dystrophin. And we decided to use CRISPR, which I'll tell you about, to correct it. Now, to put this disease in context, the first drug that was ever approved for Duchenne muscular dystrophy was, I won't get into all the details, but it's a drug that uses exon skipping. It's a short nucleotide sequence that can skip exons. And it doesn't work. And it was approved by the FDA in the most controversial approval in history in 2016. They treated boys for 1 year with this drug through an indwelling port in their chest at very high doses. At the end of 1 year, they could restore 1% of the normal level of dystrophin protein. And you have to have at least 15% of the normal level of dystrophin to be therapeutic. This drug costs $1 million a year for 1% of dystrophin. And these families are having to pay this. And there's just an absolute enormous need for something new for this disease. So we decided to try to use CRISPR. So CRISPR is a funny name. I won't get into exactly what it stands for, but there's what it stands for if you're interested. Clustered regularly interspaced short palindromic repeats. Just remember CRISPR. It's a really funny name. When you first hear about it, you think it might be like a food processor or some kind of new vegetable or something. But actually, the food analogy is pretty good because, and this really, the discovery of CRISPR highlights the importance of basically discovery-based science without a particular goal. CRISPR was discovered by scientists that were trying to figure out how bacteria could convert milk into yogurt. That's where it came from. And they figured out that bacteria in milk were turning it into yogurt, and they were studying these bacteria, and they figured out that they had this really interesting immune system. Bacteria had an immune system that enabled them to fight off viruses, like the coronavirus. And these bacteria used this ancient system, and it was called CRISPR. And then molecular biologists, including members of my lab and others all over the world, adapted this ancient immune system as a revolutionary gene editing tool to change the genome of all animals. So here's how it works. Here's the double-stranded DNA helix with the 3 billion base pairs in all of your cells. And this is a two-component system. One component is a short sequence of RNA called a guide RNA that you can synthesize, and you can make it correspond to any sequence in the DNA. So it's like a GPS device. It's a molecular GPS device, and we synthesize these in my lab all the time. There's nothing really to it. And the other part is an enzyme. It's called Cas9. That's the bacterial enzyme. And the two come together. And what this guide does is it brings the enzyme to the DNA. And when the enzyme comes to the DNA, the enzyme cuts it. And then you can edit at that cut site. So you can think about it like this. two pieces. One is the guide that we synthesize, and the other is the enzyme that came from bacteria. That's the genetic scissors. So here's what it looks like. Let's see if I can make this work.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location.
Can you hear that?
The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
So that's how CRISPR works. And now, how can you deploy this to correct a genetic disorder in people? There's two ways in principle that you could think about doing that. One is called ex vivo gene editing, and Marvin alluded to sickle cell anemia, and it has now been, many of you may have seen this in the news, it has been cured in several patients now by CRISPR gene editing to remove their mutation. And how is that done? That's done by taking cells out of the patient and then to correct them by genome editing with CRISPR, either in a virus or what's called a nanoparticle, correct the cells and then put them back into the patient, and then they basically take over in the patient. You can't do that for Duchenne muscular dystrophy because your whole body is muscle. So for Duchenne muscular dystrophy, we had to come up with an idea. How could we correct all the mutations inside the body of the person by injecting the CRISPR components? And that's called in vivo editing. So here's how it works. And again, this is, I think, my last really detailed slide. Just think about this region. Here's 4 exons. These are the segments of the DNA that give rise to the RNA that give rise to the protein. And they fit together perfectly like pieces of a jigsaw puzzle, and they give rise to the dystrophin protein. Here's the most common mutation in boys with Duchenne muscular dystrophy. They have a deletion. They're missing exon 50 of the gene. And you can see why when you're missing exon 50, you get the disease, because the exons have to fit together like the pieces of a puzzle. And 49 just goes to the next exon, and 51, its shapes don't fit. So the protein can't be made. And so this boy would have no dystrophin. That's the actual mutation that the $1 million a year drug is designed against. So we looked at this and we thought, could we use CRISPR and make a guide RNA to deliver it to this exon, and could we edit that exon, change the sequence of the exon, and either skip it, because this was really fortuitous. If you skip the 51st exon, look at 49 goes to the next one, 52, the shapes fit together. You put the thing back together like a Lego, and you can reframe the gene, you can remake the protein, or you can change the nucleotide, the DNA sequence in the exon, and you can put it back together. We call that reframing, and both of them make dystrophin. So we applied to the NIH, and we were awarded one of only 3 in the United States very large grants to set up a muscular dystrophy research clinic at UT Southwestern that I direct. And then this was our plan. We wanted to identify patients, and often we would get an affected boy with his unaffected sibling. We would take a small blood sample from their arm, 4 milliliters of the blood, and we could use a really interesting procedure to convert those blood cells into stem cells stem cells we could convert stem cells into beating heart cells. So we could start with a blood sample from a boy and turn it into his own beating heart cells in a dish. And then we could add the CRISPR components and we could ask, could we correct his cells? And if we could correct them, did they work? And this is blood sample that we converted into heart cells by this technique, and you can see they're beating spontaneously. And then we can actually put them into 3 dimensions, and we call this normal engineered heart muscle, and it'll beat. So we can not only correct the gene, we can then ask, can we make dystrophin and restore the function from this actual patient? So then came our real source of inspiration. This is a young man, his name's Ben Dupree, and he lives in our neighborhood over in University Park. He went to school with our kids. This is his mother, Debbie, and his cardiologist. So now you can see that Ben he's in a wheelchair, and he has Duchenne muscular dystrophy. So Ben was our patient 0 for testing our idea. We haven't tested it in his body yet, but we were able to test it in his cells. So Ben gave us a small sample of blood, and we asked, could we really fix his error in his DNA by CRISPR? So we took his blood and we turned it into heart cells, and these are Ben's heart cells in a dish. And then we added CRISPR to restore the dystrophin protein. And here we've stained the cells with an antibody to dystrophin in red, and you can see now his cells are making dystrophin that his body could never make, and we were able to do it with a simple gene editing approach. And this is the protein in a normal person. Here's Ben, he has no dystrophin protein, and then when we corrected it, we could restore dystrophin protein. And so here's Ben. This is one of the most inspiring moments in our lab when Ben came up to the lab and he looked through the microscope with me, and there he's looking at his cells that are making the protein that his body is unable to make.
Being able to see that gave me a lot of hope for the future, of seeing that this is a concrete thing that can happen. It could be done.
So since then, as I said, I get letters every day from mothers. We've corrected so many boys' mutations in a dish from their blood cells, and they always send me pictures. All these boys have Duchenne, unfortunately. And one of the tragic things about this disease is that it's not diagnosed until usually age 2 to 4. And there's many cases where a couple will have had a second son before the first one was diagnosed, and then they have to figure out if the second son is going to come down with Duchenne or not. So for example, these 2 boys who I know in Austin, they both have Duchenne. So there's just so many aspects to this disease. So we optimized the gene editing method using Ben's cells and the cells from all these other patients. Then was the big challenge. Okay, you can do it in a dish, but how are you going to do it in a patient or in a person? So I have a lot of really smart students in my lab we have students in my lab from a dozen countries around the world, super smart people. They all came over and we came up with these ideas. So we engineered a virus, like the coronavirus actually, but it's not the same as the coronavirus, but it's a harmless virus called adeno-associated virus, AAV. It's like a virus for the common cold, except we can make it so it doesn't cause problems. It's just innocuous. And we put inside the virus the CRISPR gene editing components, and then we built a mouse by genetic engineering that had Duchenne muscular dystrophy, and we injected it with the virus, and we asked, could this virus really find all the muscle cells in the body, and if it could find them, could it deliver the CRISPR components, the molecular scissors and the GPS device into the cell, and could it really find the one error in the DNA of 3 billion letters in every nucleus of all those tissues, and if it could it rescue muscular dystrophy? So obviously that took us a lot of potential hurdles and pitfalls along that path. So this is a histological section from a mouse. This is a normal mouse. Here's triceps, that's a muscle in the arm. Here's the diaphragm, here's the heart, all stained for dystrophin under every membrane. And here's this mouse that we made. We actually made the mutation by CRISPR, so they don't have any dystrophin at all, and this mouse has muscular dystrophy, so we injected it with the virus, and we looked 4 weeks later and we said, is it really possible that we could correct this disease in a mouse? And that's what we saw. We could we could completely restore the dystrophin protein in every muscle tissue of these mice within 4 weeks. And there's a lot of other data I'm not going to bore you with, but this was a really good day, I can tell you. They're not all this good. So then we thought, okay, we could do it in a mouse. That was really incredible, actually. But could we do it in something bigger? And so we searched the world over, and we found a colony of beagles that's maintained at the Royal Veterinary College in London. And it turned out they had the same mutation that's the most common one in boys, and that we'd already just corrected in mice. This is an interesting story. There was an elderly lady whose favorite dog lost the ability to walk and couldn't keep up with her going up the stairs, and she took the dog to the Royal Veterinary College, and they looked at the dog and they said, "We think your dog has Duchenne muscular dystrophy," and they asked if they could do a genetic test, and they sequenced the DNA of the dog, and it turned out it had the same exact mutation that caused Duchenne in the dog. So we, a postdoctoral fellow from my lab and I, we created the virus, and we flew to London, and we injected these dogs with the virus. And nobody had ever injected a large animal bigger than a mouse with a CRISPR virus. So really, I didn't know what was going to happen. I thought this could blow up on the launch pad. So that's what we did, and we did that in collaboration with Dr. Richard Piercy, who oversees the facility. So this is a biopsy from normal brothers in the litter. They all have dystrophin, which you've already seen. Here's one of the puppies that has Duchenne. He has no dystrophin. So we did this by an intravenous infusion of the virus, and we looked 8 weeks later, and we could bring dystrophin back in these dogs. That was a really good day. So here's what the dogs look like. So the first dog, the dogs are really cute. This is a normal brother dog in the litter. In a minute, you're going to see his brother who has Duchenne, and just like in boys with Duchenne, you're going to see his muscles are atrophied and he's not able to run, or he has to hop like a rabbit, unfortunately. So this dog has Duchenne, and then we treated his brother with the virus, and you'll see his brother in a moment. So he can run, he can jump. The caretakers who treat these dogs like they're children couldn't tell the difference between this Duchenne dog and a normal dog. So I could watch these videos all day. So yes, that's the first dog that's ever been cured of Duchenne by CRISPR gene editing. So that's here's where we started. We started with an idea of how we could use the adaptive immune system of a bacteria to edit genes in a human being. We obtained cells beginning with Ben and many other patients, turned them into stem cells, into heart and heart muscle cells in addition, corrected them by gene editing. We then made mice that had the same mutation through CRISPR and corrected them with the virus, and then we were able to correct dogs with the virus. And this is on the path, we hope, someday to correcting human beings. So there are many steps along the way to developing a therapeutic like this, because this has never been done. So you can imagine you got to be really, really careful. So what we're doing now is we're optimizing gene editing technology, and we're optimizing the virus technology for how to deliver it efficiently. We're making sure it's safe. We're making sure there's something called off-target. You don't want to edit the wrong gene, so you got to make sure this is really precise to go just where you want it to go. And then we're adapting it to other monogenic disorders. I told you there's 800 genetic diseases of muscle. This is a huge area of unmet medical need and a lot of opportunities. So in the last few minutes, I want to just close by telling you where I see CRISPR going and what I think this technology is really capable of. There are more than 3,000 genetic diseases caused by a single error in DNA. So 1 in 20 people has a genetic disease, and many of these you've heard of. We heard about sickle cell, Huntington's disease, ALS, cystic fibrosis, Tay-Sachs, many others. So all of these genetic diseases are ultimately amenable to CRISPR correction, to fix the misspelled letters in the code of life in the patients that are afflicted. So what are some of the applications of CRISPR that we will see eventually in the years to come? Certainly CRISPR will be deployed to correct genetic diseases. It's already been deployed for sickle cell. There's clinical trials right now to cure blindness with CRISPR by injecting it into the eye. There was a paper that came out today that's using CRISPR to lower cholesterol levels. You name it, if it's due to an error in DNA, it's probably going to be amenable to CRISPR correction. CRISPR has already been adapted to make a test for COVID-19 because CRISPR can recognize DNA sequences with the appropriate guide RNA, and it can cut the DNA sequence. And so one of the most sensitive COVID tests is based on CRISPR. It's being used by scientists to eliminate malaria so they can infect malaria-carrying mosquitoes with CRISPR, and they can cut out the infective gene in those insects. It's being used to create to kill antibiotic-resistant bacteria. It's being used to enhance crop production and to make stronger livestock, super cows, super milk producers, etc. CRISPR has been used now to make seedless tomatoes, gluten-free wheat, my wife would like that, roses that will never wilt. All of these things, if they're dependent on a DNA sequence or a gene, they can be corrected by CRISPR. I'm on the advisory board for a large genome institute in California, and they're trying to use CRISPR to prevent global warming by affecting plant emissions and anything else you can imagine that has to do with DNA in an animal or a plant or a bacteria or a virus can be addressed by CRISPR. What are the perils of CRISPR? Germline editing in humans. When CRISPR first was described, many of us scientists thought this someday some rogue scientist might use this to try to change the genome of human beings. And within 2 months after CRISPR was after that started being conceived of, many of you may have read, Chinese scientists created CRISPR babies. There are 3 babies born now in China whose genes were edited by CRISPR, and that was extremely reckless because there was no regulations or oversight on that. But that changing genes in people through what's called germline editing, I should probably clarify this. Germline editing is a form of editing where you change the eggs and the sperm in a person, and so whenever they have offspring, that change that you introduced will be passed on through the gene pool. The gene pool is like a river that flows to the future, and if you make a change, it flows to all of subsequent offspring. That's different from what we're trying to do with Duchenne boys. What we're doing with Duchenne boys is to change the DNA in the patient, and that therapy is confined to the affected patient. It's not passed on to their offspring. What's another peril of CRISPR? Editing errors. You may intend to target a disease-causing gene, but what if CRISPR is not 100% perfect, which it's not, and it hits some other gene that can cause cancer or it can cause some other disease. So you've got to make really sure that this is perfect. And bioweapons, it's being used, it's already being used, we think, to develop bioweapons that could make a virus with some toxic CRISPR and infect communities. It could happen. The ethics, what are the ethics of CRISPR? Should we improve the human race? If so, who decides on the ideal traits? It's one thing to get rid of a disease like Duchenne, but it's another thing to decide you want your child to have blue eyes or bigger muscles. And who gets access? It's expensive. Does affluence equal access? And what about, should we eliminate undesirable species? You never quite know the consequences. Okay, you could eliminate mosquitoes because you don't like mosquitoes, but once they're gone, then you might find out, well, that just disrupted the whole ecosystem. Or should you create super species? Again, there's a lot of issues, and this is going to require really careful dialogue between scientists, bioethicists, governmental people. Scientists alone aren't qualified to make these kind of decisions. Scientists, we're just figuring out what is possible and figuring out the rules, but it's not up to scientists to really decide how these technologies are deployed. So lastly, I was looking up in the sky in South Dallas, and I saw this cloud. I don't know what that looks like to you. To me, it looks like a double-stranded break in a double helix of DNA. So I sent it out on Twitter. It went viral on Twitter. Spontaneous DNA cloud above double-stranded break, with double-stranded break above Dallas. A good omen for CRISPR gene editing. And here's a close-up. See, that's not Photoshop. So anyway, I thought that was a really, really good omen. So this work would not have been possible without a great scientific team and a lot of support, but really the support of the Duchenne community has really enabled us to test our ideas. This is just one meeting in Austin. All these boys have Duchenne and their families, and they're really just so incredibly inspiring. And they really have provided us with a lot of inspiration, enthusiasm, and whatever we've needed to try to move this forward. And usually every morning when I get to work and I have letters in my email, I forward them to the members of my team so they can feel that they're really part of this. There's also been a lot of community support for what we're doing, and there's a lot of familiar people here. Here's my friend Willie and his wife, Annie. Here's our governor and former president, and Dateline, we're twin brothers separated at birth. That's Keith Morrison from Dateline NBC. So anyway, there's been a lot of excitement about what we're doing. So this is my current team at Southwestern. Here's where we're located on the North Campus at UT Southwestern. The work, to transition this work from an academic lab toward a clinical program requires a company. You can't really do this with a bunch of students and kind of chaos in an academic lab and thinking about things. You really got to be organized. So that's why we founded Exonics, and then that was acquired by Vertex, which has taken this forward in a very large facility in Boston. And our Muscular Dystrophy Research Center at UT Southwestern has really enabled this. So that's all I've got, and I'm happy to answer any questions.
How close are you to using this in the patients?
So that's always the hardest question, for sure. The only challenge that stands before us right now to move this into humans is the manufacturing of the virus. So that's what we're doing. Everything else is optimized and works really well and is safe, so we have to make enough virus to treat all these boys, and that's a manufacturing challenge that we're working on. So our hope is that we will be ready for first-in-human trials by sometime in 2022. We hope. That's what we hope, but we'll have to see how things proceed. So safety is paramount.
So does the gene repair persist in those beagles, or do they lose it?
Great question. So she's asking, does it last forever? So it does last, as far as we can tell, at least in animals that we've looked at so far, it lasts forever. And the reason is because heart muscle in particular lasts forever, as I mentioned. So once you correct it, it should be sustained. And we think in all the skeletal muscle fibers, it will also be permanent as far as we can tell.
But there's no immune reaction to the dystrophin.
That's a really good question. So that relates to all gene therapies. If you're putting a missing gene back, do you get a immune response? Without getting into too many details, there is not an immune response to dystrophin, and that is because boys who have Duchenne have what's called revertant fibers. It's like 1% or less of their fibers actually make dystrophin, a really small amount. It's not enough to benefit them, but it's enough so that their immune system has been exposed to dystrophin. And so if you then put the normal level back, it's not recognized as foreign, and so there's not going to be, we don't think there's going to be an immune response. Now that said, CRISPR will likely elicit an immune response because it's a foreign protein, and so probably this will be delivered together with immunosuppression to prevent an immune response, at least initially.
Did you see in the dog any, during the 8 weeks, any symptoms?
Now we looked at that. We looked at T cell responses and other immune responses, and we did not see any at that time. And half the dogs received immunosuppression and half didn't. So I think that's hopeful news, but I think we need to do longer term studies.
How close to normal do you think these kids will get after treatment if it's successful?
I think it depends when the therapy is delivered. So the earlier the better. Based on our animal studies, if you deliver early while you still have muscle function, you can preserve that. If you wait until too late and the muscles are already deteriorated, there's nothing left to fix. So we really want to go as early as possible, and we think from the time of intervention forward that they will be largely corrected. That's the hope.
Can you tell us simply how the virus you inject with a code knows where to go to?
Yeah. So we use a type of virus called AAV9, adenovirus 9. It's safe. It's been used in a lot of gene therapy trials. It's already approved by the FDA. And there's one form of AAV called AAV9, and on the outside of that virus, there's proteins. You've heard of spike protein for coronavirus. I think we all know what that is now.
Been vaccinated.
I hope everybody in here has. So have I. There's a protein on the outside of the adenovirus that we've injected that goes right to muscle and heart and nowhere else, and it's very specific, and it's fortuitous. That was found by screening hundreds of viruses, and it, for whatever reason, that makes a protein that just has a high affinity for muscle. So that's what we use. It's almost like a specific delivery system for that tissue.
But I mean, other things that you might correct. I mean,
So there are other, as I mentioned, we're using AAV9. There's AAV1 through however many you might think about. And another company that I founded, it's called Tenaya Therapeutics. It's based out of San Francisco. And we have a complete manufacturing division in the company, and they're screening for all the different types of viruses that can target different tissues, like selectively tissue the heart or selectively tissue nerve cells. And so those will be, we hope, someday you could take the virus, if you've got a disease you want to correct in the brain or the liver or the kidney, you would get the appropriate virus and put the CRISPR components in it, and then you would use that as the tissue-specific delivery vehicle.
Is that just a striated muscle? What is the effect on smooth muscle?
The two major muscles affected are striated, skeletal, and heart. There's not as much effect on smooth muscle. There may be a subtle effect, but right now we're going for the big problem, which is the heart and skeletal muscle.
If I understood you correctly, this correction that you showed took out another section of information for
Yes.
these payers. and left it out, and so the dystrophin is not perfect, but it's usable.
Yes.
Now, is that the same situation in all the dystrophy cases that you said there were dozens of different mutations? Is it the same in all of them? Same situation?
It's a good question. Most of the dystrophin mutations involve deletions, where you're taking a segment of the gene out. And if the segment of the gene is taken out, you can't easily put it back. There are ways to do that, but that's a whole nother lecture. So what we're trying to do is just take the remaining part of the gene that the patient has and fix it, put it back together as best we can. So it's, as I mentioned, it's like a shortened shock absorber. It's not perfect. It's like putting a spare tire on your car. It'll work, but it may not be perfect. But compared to what they're dealing with nothing, it's a lot better than that.
So will they be like Becker's?
They're going to be mild Beckers, or better than mild Beckers, but it's going to depend on what their deletion is. Yep. I mean, the huge goal in the DMD field has been to convert DMD to BMD. If you could take somebody and let them be able to walk around till they're in their 70s versus being in a wheelchair at age 10, that's a big difference.
Did your treated dogs have normal puppies?
We have not bred those dogs, so I don't but their puppies would not be affected by the approach we took because, remember, we didn't do germline editing. We're not affecting their sperm or their eggs. We're only affecting their tissues. So if a affected person that we corrected had children, they would be affected. But without getting too complicated, the mutation is only passed from the mother to the and we're only treating boys. So
are you able to do it in utero? You could screen the dogs, and 50% of the males are going to have the disease from that colony. What if you did it in utero and tried to do the gene editing before they were born? I guess you'd have to do it after the cells differentiate.
Well, if you could do pre-implantation genetic diagnosis and identify probably Duchenne, but editing it in utero is tough. And the other problem with finding mutations in this gene is that the gene is so big. Until you know you got a mutation, it's kind of hard to find it.
Are you at present working on any other disease specifically?
Yes, it's a good question. We're working on a whole series of cardiomyopathies. So there are a number of hypertrophic and dilated cardiomyopathies that are caused by well-known mutations in proteins involved in contraction, and we are working to correct a number of those right now. And we're working on even a more precise, I didn't get into it today, but there's an even more precise form of gene editing where you can go in and you can change one letter in the DNA. If it's an A and you want to change it to a T, there's 4 letters in the DNA code, A, C, G, T. And you can actually, just like a spell checker, you can change one to another now with this gene editing approach.
I like it.
I had a son that died from a mutation, a genetic mutation, a disease that was caused by that. And really what you're doing is more than, as much as eliminating the disease, is eliminating suffering. And I think that word really is the key or the motivation behind what you're doing. But I was curious, I couldn't understand, it's like if you do this on one person and it helps, by whatever standard you decide that it helps. And I can tell you from my experience, if you have someone that, like you're saying, would die very young, you could ask them 2 or 3 weeks before that, or when they start getting really bad, will they take a chance on that? I don't think any of them would refuse. So, so you have, once you do that, is that the same procedure that will be used for every other disease, or is there going to be a learning curve for every disease that you treat? Does that make sense?
Yeah, it makes sense. You have to, The GPS device, the molecular GPS device, that's the guide sequence that you have to program to recognize the target sequence. That's going to be different depending on whatever mutation. You can deliver it wherever you want, but the enzyme that actually does the edit, the molecular scissors, that's the same. So what we're trying to develop is what you would call a plug-and-play platform where you just swap in or out whatever the guide RNA is for the sequence you want to edit, and you don't have to rebuild everything. You're just swapping in the one component.
So, shifting gears a little bit, instead of correcting genetic deletions, how would you go about selecting the appropriate place for genetic enhancement? For example, you showed the picture of the hummingbird, so contractile enhancement or grasshopper muscle or something like that. Where would you determine to place that in the genome?
Well, if you know the gene you want to target, so there's one gene that controls muscle mass, and there's been spontaneous mutations in this gene. It codes for a protein that's secreted, and if this gene is mutated, it causes super muscling. Like there's cows that have 3 times more muscle, and it works in all animals. And there's people now that are trying to say we should we should mutate this gene in humans and turn everybody into Arnold Schwarzenegger. So there are ways to do enhancement. They think there's ways to improve your eyesight by you can not knock out a gene, but you can change the sequence of a gene to make it better. So
volunteer.
So there are definite ways to do genetic enhancement based on the knowledge. You have to know the gene that you want to target, and you have to know how it works. But if you know that and you know a way to make it work a little better, you can in principle do that by CRISPR.
And you could do that with species variability.
You can change species for sure. I mean, that's, as I mentioned, that's being done. It's already being done by in mosquitoes, where they gene edit mosquitoes, and then they turn them loose into the mosquito population, like in Africa, and they can make like the males sterile, so they'll mate, but they can't give offspring, and they can also put in suicide genes that'll kill them. There's all kinds of. It is science fiction, really, but it's becoming reality.
I've got a question.
Oh, my wife has a question. Is it scientific?
Well, it's clinical. I'd like to know if someday are they going to have, when newborns get their heels pricked and they fill 5 spots of blood on the paper to test for thyroidism, hypothyroidism, all those inborn errors of metabolism, taste, PKU and all that.
Yeah.
Is there going to be one for muscular dystrophy someday?
So that's a good question.
Say that.
You could. I better say that. That's a great question, honey. No, there's. You can already test for Duchenne in newborns. It's a simple blood test, and many of the clinicians will know this. You test for creatine kinase, which is a muscle enzyme in the blood. If it's elevated tenfold, it's diagnostic for Duchenne.
Are they doing it in all 50 states?
2 states do it.
Ohio, I bet.
Ohio's one, and I forget the other one, but they offer it. But the majority of families don't want the test.
Why? Because the earlier they're
Because there's
diagnosed,
nothing they can do if they're. I'm not a clinician. This is what my clinician friends tell me, that a lot of families don't want to do the test because what are they going to do with the information if it's positive? They'd rather be blissful. But it is available for testing just by the simple CK creatine kinase assay.
But you would be in the future treating babies, not 3-year-olds.
Ultimately, you'd want to treat people at the time of diagnosis at birth. But yeah, that's the hope.
Are most of these picked up in a genetic testing? They do you have your genome done? Have you had that done?
I've had my gene. I've done 23andMe, but I haven't had my whole. sequence. All these Duchenne cases are, basically all of them, are not, the family doesn't even know they have it until the child is born. And another challenge with prenatal diagnosis or even family testing is that 1/3 of the boys with Duchenne inherit the mutation as a spontaneous mutation because the gene is so big, it's so complicated, and a lot can go wrong when you got a gene that's 2.5 million letters long. So 1/3 of these boys, so the mothers aren't even carriers. Something happened during early development and they got a mutation, and then the son got it. And then 2/3 are genetic carriers.
If that's the case, then these specific areas you mentioned must be areas that are prone to breakage or malfunction. Is there anyone working on trying to understand that or prevent that?
Yeah, so you're absolutely right. Those hot spots is what I call them. There's 79 exons, but 80% of the mutations cluster in 10 of the exons. So those are the genetic hotspots. And we don't really know the reason, but probably the DNA or the sequence is kind of fragile right there and may have a tendency to recombine or not replicate correctly. So there are people working on that. We're not working on that ourselves. We're just focusing on the areas that we know are mutations, but that is a big issue.
Can CRISPR work be applied towards dwarfism, and has any work been done with that?
It can certainly be applied toward dwarfism. The clinicians will know more about this than I will. I think dwarfism is frequently caused by a mutation in the FGF, fibroblast growth factor receptor, FGF receptor, that's homozygous, meaning 2 copies are mutated. So knowing that error, that would be readily correctable by CRISPR.
At birth, or when would
I don't know. You could correct it at birth. I don't know whether the dwarfism is already heavily manifested by birth. Obviously, it's due to growth plate closure issues. Wow, okay, you guys are a great audience. Thank you so much.